Hi there, and welcome to an overview of the specific immune system. The specific immune system refers to the lymphocytes that target particular antigens. So this differs from the nonspecific system, which is a much more generalized response to any antigen. So kind of the beginnings there is the presentation of an antigen that comes in. And we'll kind of highlight both the B and the T cell, but there is some overlap between the two, so I'll be drawing them in together. So the antigen is first thought to be taken up by one of the monocytes in most cases. Now you can have direct antigen stimulation of the B cell, and there has to be some antigen stimulation, but generally the T helper cell comes in. So I'm going to kind of start that pathway a little bit first. So I have drawn here a dendritic cell. And the dendritic cell acts as what we call an APC, or antigen presenting cell. A lot of terminology here, so I started with a few notes. I'm going to add a few more here as we go. Whoops, antigen. Okay, and they're going to ingest and then present the antigen. So we'll just make the antigen kind of a shape here. So this is what I'm going to use here as an example is a bacterial cell is fine. So we'll call this our antigen. But remember, this is anything that the body recognizes as not self. It has a much broader definition. So it's taken up, possibly phagocytized, hopefully phagocytized, and then it's presented on the outside of the cell with things called MHCs. And one thing to note when we think of an antigen and we're talking about a bacterial cell, we think of maybe the whole cell being presented, but in fact that's not really what happens. The antigen, the part of it that stimulates the specific immune system are fragments of that, and there's specific regions that invoke this immune response. So those areas are called epitopes. But often you'll kind of hear or read that the term antigen is used kind of generally and used interchangeably with epitopes, but epitopes are really just a small piece of the antigen. What's nice about that system is that you can have an antigen, a bacterial cell, and have multiple epitopes, different epitopes, so you can have multiple stimulations in the process, which just then further kind of accelerates and broadens the attack on the whole antigen itself, because if the B or T cells are acting upon different parts of the antigen, it's just more areas kind of to attack. Okay, so initially you're going to have binding to a couple different possible T cell types. So I'm going to start with the T helper cell. And shorthand is for that. So typically in the beginning we call it either an immature or a naive T cell. Sometimes it's listed as effector T cell, so it really refers to the fact that it's this immature, not yet stimulated T cell type. So if it's going to become a T helper cell class, there's receptors on there called CD4. The T cell type will have these areas called TCRs, or T cell receptors, and that along with the CD4, which kind of wraps around here, bind to this epitope, and then it will be stimulated to divide. Now when it divides, it can become different types of we'll put active or mature. And there's different types of T cells, or T helper cells. There's different subtypes, and they have different functions, different stimulatory functions, but in general they stimulate different white blood cells. So that's kind of their big picture activity that's there. And the compound that they often use to do that is called interleukin. And interleukin is going to be really important to stimulate the B cells as well. And again, there's different interleukin types, so we're just going to kind of stick with the major groups that are here. Another type of T cell that can bind as well are the CD8 cells, which are going to be activated to become cytotoxic T cells. So those, let's come back to that. We'll do the B cells next. So as this interleukin is secreted and this T cell becomes activated, it can now bind to, so the T helper cell binds to, again, this kind of immature, sometimes it's called effector, B cell. Now the B cell itself, in order to be activated, it's kind of a multi-step process, and this activation process is really important because you don't want to activate these cells unnecessarily and run the risk of producing either cytotoxic T lymphocytes or antibodies that attack, attack either self cells or, kind of attack indiscriminately. So maybe attack your normal microbiota or attack food that you eat. So there needs to be these kind of multi-step activation processes to provide some control. Now the B cell, we'll call it immature, and again there's different terms, also is going to interact with the antigen on its BCR. Get our antigen back over here. And then ultimately it's going to be completely activated by this kind of second tier step, this T helper cell binding to it and then that secretion of interleukin. Now there are some antigens that are sufficiently small that they can stimulate the B cell directly, but typically you need the addition of the T helper cell, and hence the name HELP. So when this matures, it's going to form a couple key types. There's going to be BM or B memory cells. There's going to be B regulatory cells. Sometimes those are referred to as suppressor B suppressor cells because the idea is that it'll dampen down the response, like when enough is enough, the antigen's cleared out and you kind of want to shut the response down. And then the one that we get really excited about as well are the B plasma cells. And the B plasma cells are the antibody secretors. And I'll talk more in a different video about the functions of antibodies, but we'll just kind of leave it now just to say that the antibodies are proteins that bind to the antigen directly. And that's what's kind of special about antibodies, is that they bind to the antigen itself. And they're typically shown as Y-shapes. Sometimes they're multiple Y-shapes linked together, depending on what kind of antibody type you have. But the general base structure is the Y-shape itself. Okay, so that is the B cell pathway, and this is what we typically refer to as either B cell or humoral immunity. And that's because antibodies circulate in the blood or the serum. So the humerus was kind of the general term for body fluid. So because the antibodies themselves circulate in those fluids, it's been called humoral immunity. So the T cell, let's go back to our T cell. So we talked about the stimulation of the T helper cells. Also some other T cell types that are stimulated are the T regulatory cells as well. And then this is going to help stimulate another type of cell. So this is not going to mature into a cytotoxic T lymphocyte, but instead you're going to have binding here, and I'm going to draw this a little bit small to kind of squeeze it all in. So you can have these kind of immature CD8, which refers to a different type of receptor. So it's another kind of T cell, immature or effector. It's not quite matured yet. It also will bind to the antigen presenting cell. And of course, there's going to be multiples of these. And it's also got a, let's keep the same color. It's also got a TCR, but that wraparound protein is the CD8 instead of CD4. And it would bind to an MHC. I'm going to just squeeze an MHC in there with epitope. Got to have that epitope presented. So what's happening here by the presentation of the epitope? Well, you have many different types of B and T cell subsets. So, okay, we're talking about these major groups here, but then you have different types of those that respond to particular antigens. And these form early in development. They form through gene rearrangement and all these different possible cell surface markers that are on the outside. So we're selecting very specific, not just types of B and T cells, but subtypes of that are specific for that epitope or that antigen, if you want to use the term loosely. So now this CD8 cell, so it's not necessarily connected to these two, except I did want to point that out because the interleukin is going to help that out. T helper cells are so important there. There's our interleukin again. And again, there's different types of interleukin, and a lot of that refers to which to the white blood cell type it stimulates. And now this cell is going to become what's called either a cytotoxic T cell, but the kind of true active term is cytotoxic T lymphocyte. And I'll put that over here. This is the active cell destroying T cell type. I'm going to squeeze in here cytotoxic T lymphocyte. Okay, so this CTL now is actually going to attack infected cells. So it's going to bind to So we'll actually seek out cells that are also presenting on their MHCs pieces of the epitope or some irregularity there that signals that particular microbe has infected that cell. And it destroys it through a couple processes here. It basically punches holes in the membrane and then allows materials to leach over so that the membrane ruptures. So it's going to cause infected host cell lysis. And it secretes these two major groups of enzymes, the perforins and granzymes. Let's try to squeeze that in there. Okay, a lot to squeeze in here. So they'll secrete these compounds and cause those cells to rupture. Now, another really important piece here is that you're going to get T memory that are going to form. And that's going to come from that CD8. So you get T memory forming as well. Okay, so let's talk just briefly about memory. So memory refers to, you have these subtypes already present, but once you've been exposed to the antigen, that you form these sort of kind of latent cells, but they can become active very, very quickly. So when you're re-exposed to that same antigen, they'll quickly jump to work. You don't have to go through quite as many steps as these activation processes, and ultimately reduce infection and then the presentation of disease symptoms in the host. So this is the whole idea behind vaccinations, is that we stimulate these processes here without necessarily developing the disease and having the symptoms. And sometimes you talk about things, certain vaccines like mild fever, or you might have rash or something at the site depending on what the vaccine is. So you may have some mild symptoms symptoms of that, but you don't get the full-blown disease. But with the vaccines, the idea is even though you're not getting all those symptoms, you're developing memory. And so that when you do get exposed to an active, virulent form of that particular microbe, that those memory cells jump into action so quickly that they help eradicate it, whether through stimulating phagocytosis, whether it's through attacking host cells, producing antibodies, they help eradicate it so quickly that you don't develop a full round of the disease. Okay, so I'll talk about antibody function a little bit later on, but this is just kind of an overview. Quick recap again. So when the antigen comes into the body, you have different monocytes. So you can have macrophages, you can have macrophages, and you can have B cells, dendritic cells, histiocytes that will all bind to it. And by doing that, you end up stimulating the T helper cells. And those will secrete interleukin. And then with the B cells, they'll bind to active T helper cells, also picking up that interleukin that's there, and then quickly become active to make antibodies and make memory and regulatory cells as well. With the cytotoxic T cell, it's a different precursor type. So we have that CD4 that becomes the T helper cell and T regulatory. And then it's a different precursor type. It's a CD8, but it's related to this whole class of T cells. Now this will become activated to actually go and go and attack infected host cells. So important, particularly in viral infections, where if you can halt that multiplication process pretty quickly, then you can keep that virus from making copies and spreading. Okay, that's all I have here for this. Please contact me if you have any questions. This is Corey Andres with C&M Glass.